They told us the Maya disappeared, swallowed by jungle, scattered by conquest, erased by plague. Cities like Copan, once home to 20,000 souls, emptied by the 9th century, their writing stopped, their gods forgotten. And yet, in 2025, deep inside a lab analyzing ancient remains from Copan, scientists ran a test no one expected to pass. 7 individuals, buried over 1,200 years ago during the classic Maya period between 600 and 850 CE, had their genomes sequenced, strand by ancient strand. What came next stunned everyone. The DNA matched. Not approximately, not distantly, but directly to living Maya populations today in Guatemala, Chiapas, and the Yucatan. These were not ghosts. These were great-grandparents. For over 9,000 years, the Maya genome had survived. From early Holocene settlers around 9,600, 7,300 calorie. BP, through the rise of city-states, the Spanish invasion, forced Christianization, and the collapse of dynasties, their blood held the line. People's history declared extinct. Never disappeared. How is that even possible? How did a civilization facing deforestation, megadroughts, the fall of over 40 city-states after 900 CE, and smallpox waves in the 16th century still maintain their genetic identity? Even after 1524, when Pedro de Alvarado's campaigns devastated the Guatemalan highlands, even after millions were lost, the DNA persisted. Modern Mayas still carry mitochondrial haplogroup A2 at astonishing levels, 77.3% in Quintana Roo, 98.8% in the Lacandon of Chiapas. And the paternal Y chromosome? QM3, the Pan-Amerindian marker, unchanged since the original migration from Siberia some 15,000 years ago. But here's the twist. The DNA wasn't just preserved, it was different. Unique internal frequencies. Rare HLA immune alleles like DRB10407 and DRB10802 appear in over 50% of Maya populations. Combinations of mtDNA haplogroups never seen elsewhere. This isn't just continuity, it's defiance, coded into the genome. And buried even deeper is a mystery the data couldn't explain. Because some of those ancient Copan skeletons carried markers not found in today's Mesoamericans. Traces of rare lineages D4, H3, A5, and C1b linked to ancient, nearly forgotten migrations. So now the question isn't just how the Maya survived, it's who came before them. And to answer that, we must follow a trail, back through the smoke of time, to the very first humans who set foot on this continent. To answer that, we have to leave the pyramids behind and step into a frozen world. Around 15,000 years ago, long before the first Maya word was ever spoken, the Earth was thawing out from the last glacial maximum. But one narrow path remained: Beringia, a frigid land bridge stretching between Northeast Asia and the tip of Alaska. It's here the story of every indigenous American begins. These were the first peoples, carriers of the founding maternal haplogroups, A2, B2, C1, D1. Mutations passed from mother to child, generation after generation, tiny molecular signatures now scattered across the continents. And yet, the Maya didn't just inherit these haplogroups, they reshaped them. Thousands of years later, in modern Maya populations, we find A, 2 dominating places like Chiapas, nearly 99% in the Lacandon. B, 2 emerges strong in the Tojolabal. C1 scattered across ancient burial sites, and D1 often absent but once something else. Because between 9,600 and 7,300 calibrated years before present, the early Holocene, the Yucatan Peninsula, held people whose DNA doesn't fully match any modern Maya group. In a pre-Columbian burial from Quintana Roo, a young woman was found carrying D4, H3A5, a lineage now nearly extinct in Central America. Another, from the same period, carried C1b. These aren't common Native American signatures. They're rare even in early Holocene remains. And most critically, they show only distant genetic affinity to present-day Maya. So what happened? If these weren't the direct ancestors of the Maya, then someone or something must have come after. A new group, a new migration, a replacement or mixture so dramatic, it shifted the entire genetic structure of the region. And when it came, it brought more than genes. It brought agriculture. It brought maize. And according to ancient genomes dated between 5,600 and 3,700 calorie BP, it brought a new ancestral source. One related not to northern migrants, but to something few expected. The Chibchen peoples of southern Central America. A population that would eventually make up over 50% of Maya ancestry. So now, we follow the trail south to find the ones who crossed rivers, carved through jungles, and carried the seeds that would reshape the Maya forever.
A population that would eventually make up over 50% of Maya ancestry. They didn't come from the north. They came from the rainforest. Between 5,600 and 3,700 years ago, long after the glaciers melted and long before the rise of Tikal, a new migration crept northward, not with spears or conquest, but with maize and memory. They were people related to the Chibchan language families, a vast web of cultures stretching from northern Colombia to Costa Rica. And though their names are mostly lost to time, their genetic footprints remain. Not in artifacts, not in ruins, but inside living Maya blood. The science is clear. Ancient genome sequencing shows that 52% of the modern Maya genome descends from this wave. A people once unknown in Maya history, now proven to be its backbone. And they brought more than DNA. They brought maize horticulture, not the wild teosinte grass of ancient foragers, but fully domesticated, high-yield maize, a crop born of careful selection, a crop that demanded land and fire. This moment left visible scars. Charcoal traces in sediment. Pollen shifts in soil. Clear signs that forest after forest was burned, tilled, and reborn. And with each flame, their legacy grew deeper not just in the landscape, but in the genome. Mitochondrial haplogroups C, 1, C, and A, too, surged in frequency. The older D4H3, lineages from 9,000 years earlier, began to vanish. The ancient ghost population faded, replaced, reshaped, rewritten by the fire farmers of the South. But the real mystery is what happened next. Because somehow, in the centuries that followed, despite droughts, invasions, and collapse, this new ancestry didn't fade. It didn't dissolve into history. It held strong. In fact, it strengthened. So now we turn to the next puzzle. How did this new bloodline remain so untouched, so tightly preserved, when the world around it changed beyond recognition? The answer lies not in the jungle, but in the village. Not in walls or weapons, but in the quiet decisions made across kitchen fires, across generations. Endogamy. For thousands of years, Maya communities didn't just build cities, they built barriers around their blood. Invisible, cultural, fiercely local. And over time, those decisions carved something rare into their DNA, genetic continuity so deep, it defies the chaos of history. By the 20th century, this wasn't theory, it was measurable. Studies show that more than 80% of Maya individuals were born and married within their own communities, creating a closed loop, village to village, family to family, decade after decade. And while the world modernized, something unexpected happened. Between 1,937 and 1,969, fertility rates among Maya women peaked at 7.2, 4 children per woman, a demographic surge that flooded their lineage forward. Even in the following generation, between 1,970 and 2002, fertility remained high at 4.00 children per woman. This isn't just a population boom, it's a genetic echo chamber. Nowhere is this more extreme, more haunting, than among the Lacandan Maya of Chiapas. In a modern genomic analysis of 81 individuals, 98.8% carried mitochondrial haplogroup A, no B, no C, no D, just A. Their genetic diversity score? 0.025. That's not low, that's near silence. This is what happens when a community closes itself off for centuries, geographically, culturally, and reproductively. The Lakandan didn't just inherit the past, they sealed it. But here's the twist. Just beyond their forest walls in the same Chiapan highlands are Maya communities that tell the opposite story. The Toholabal, for example, carry 58.1% haplogroup B, a lineage rare even among neighboring groups. The Tzotzil, a quarter of their lineages belong to haplogroup C. And the Quiche of the southern highlands? They have no haplogroup A2 at all. So if the Maya preserved their ancestry through unity, how do we explain these radical genetic fractures? What happened in these bloodlines that caused them to rewrite the rules? Because not all Maya followed the same path. So if the Maya preserved their ancestry through unity, how do we explain these radical genetic fractures? What could splinter a bloodline so consistently preserved into such sharp internal contradictions? Because just beyond the Lakandan rainforest, where 98.8% of people carry a single mitochondrial lineage, the genetic map explodes into something else entirely. Let's start with the Toholabal Maya, also in Chiapas. At first glance, they share the same landscape, but inside their DNA, the story flips. Only 25.7% carry haplogroup A, the very lineage nearly universal among the Lakandan. Instead, 58.1% carry haplogroup B, a lineage more common in South American highlands than Mesoamerican forests. And C? Completely absent.
No trace, as if a whole ancestral thread never reached them or was erased. Then shift slightly west to the Tzotzil, still in Chiapas. Their genome doesn't reflect the Tojolabal at all. Instead, it shows a balanced triad including 25.3% haplogroup C, the very lineage missing in their Tojolabal neighbors. These aren't minor fluctuations, these are population-level shocks. And when we travel further, into the Guatemalan highlands, the contradictions deepen. The Quiche Maya descendants of powerful post-classic kingdoms carry no haplogroup A2 at all. Instead, their genomes reveal elevated levels of haplogroup D, often associated with early North American Paleo-Indians. This isn't regional variation. This is foundational divergence. Then, something even stranger. In the Chorti Maya of southeastern Guatemala, a group that still speaks a direct descendant of classic Mayan language, we find a genomic anomaly. Not maternal this time, but paternal. Y chromosome analysis reveals that 26.3% of Chorti males carry European or African ancestry, a marker of colonial disruption, conquest, and forced mixture, yet embedded in a people often viewed as carriers of pure classic Maya legacy. Bloodlines, rewritten by violence. So now, the Maya identity isn't a single thread, it's a web of overlapping histories, broken timelines, and silenced migrations. But the deepest riddle is still ahead. Because inside one Maya subgroup, scientists found a rare genetic signal, a maternal lineage not seen in any nearby indigenous group, but found thousands of kilometers north in the Arctic. How did that get there? A maternal lineage found in the Arctic suddenly showing up in the blood of a Maya group. How could two worlds, separated by thousands of kilometers and thousands of years, be tied together by a single strand of DNA? To find the answer, we go back to one of the great cities of the ancient world, Copan, a jewel of the classic Maya age. Carved stone temples, celestial calendars, kings who trace their ancestry to the gods themselves. Between 250 and 900 CE, this place was alive with art, with power, with belief. But when scientists opened the tombs beneath its temples in the 2022nd, what they found wasn't myth, it was something far more human. 7 individuals buried between 600 and 850 CE had their genomes sequenced. The result? 88.9% carried haplogroup C. The remaining 11.1% carried D. That's an astonishing concentration. In modern Maya populations, haplogroup C rarely reaches such heights. It's often present, but never dominant like this. It means something fundamental changed. Not in who they were, but in how their DNA spread. Even more surprising, there was no genetic wall between rulers and commoners. No sign that elites bred apart from their people. Their blood was shared vertically, across classes, horizontally, across lineages. It's a truth that undercuts centuries of speculation about divine bloodlines, sacred kingship, chosen heirs. In Copan, even the powerful were still Maya. Then came the collapse. Around 900 CE, Copan fell. Its plazas emptied. Its carvings cracked beneath rain and time. For centuries, we thought that meant erasure. But the DNA tells a different story. It didn't vanish. It moved. Those lineages, especially haplogroup C, didn't die in Copan. They dispersed into nearby valleys, into smaller villages, into bloodlines still alive today in parts of Honduras and eastern Guatemala. This wasn't extinction, it was migration. But inside that blood was something else. Because while the culture collapsed, the genome didn't just survive, it adapted. And deep inside that adaptation were immune codes. Markers of resistance, of trauma, of survival written at the cellular level. So if their temples fell, but their bodies lived on, what else did the Maya carry hidden in their blood that no empire could destroy? The answer wasn't carved in stone. It was buried in blood. For centuries, we searched for Maya greatness in their cities, Copan, Palenque, Tikal. But the real story? It was always inside them, hidden in their cells, waiting in silence. The Maya immune system isn't just a defense mechanism, it's a time machine, a record of every battle they've ever fought and survived. Inside it, scientists found something almost impossible. 2 genetic signatures, DRB1x0407 and DRB1x0802 HLA alleles that control immune memory. And among the Maya? They're everywhere. 38.6% of Maya today carry DRB10407. Nearly 19% carry DRB10802. In the world of human genetics, those are massive numbers. So massive, in fact, they don't exist like this in any other indigenous population in the Americas. But where did they come from? Some say it's the jungle. 1,000 years of surviving parasites, dengue, rotavirus, a biological pressure cooker. Every fever, every wound, every death reshaped their genome, one mutation at a time. Others see something stranger. Because those same alleles DRB1x0407 and 0802 show up far away. In Siberia. In Arctic peoples. As if some ancient echo, some shared ancestral fire, still flickers between ice and rainforest. But it doesn't end there. After 1492, the apocalypse came. Not through war, but through skin, smallpox, measles, influenza pathogens with no mercy. Entire nations were erased within decades. But the Maya, some communities didn't vanish. They endured. And now, one theory has taken shape. Gene conversion. A desperate genetic reshuffling after European contact where immune alleles merged, mutated, adapted in real time, creating something that wasn't there before but had to exist, or nothing would survive. A hybrid immune code, born in crisis, sculpted by the empire. Not all Maya made it through, but those who did carry more than memory. They carry molecular weapons evolved in darkness. And that leads us to the final twist. Because in one isolated Maya group, these immune codes are paired with something else. A signal so old, so rare, it doesn't belong to Mesoamerica at all. It belongs to the very edge of the continent. But how did that gene survive when every map said it shouldn't exist? To understand how that fragment survived, we need to go back not to the height of Maya power, but to its unraveling. Between 700 and 900 CE, the classic Maya world didn't collapse with fire and conquest. It withered, slowly, mysteriously. The king still ruled, the temples still stood, but something deeper broke. Droughts deepened, crops failed, water tables dropped. Cities that had once charted the stars began crumbling from within. Tikal, Copan, Calakmul, gone. And just when their world cracked, the next storm arrived. By the early 1500s, Maya civilization faced something it had never seen before. Steel, crosses, horses, disease. Smallpox hit before the first musket fired. It didn't just kill, it erased. Entire communities vanished without a fight. Villages emptied in weeks. But in the ruins, something strange happened. Some groups didn't scatter. They closed in. Geneticists call it a bottleneck when a population shrinks so sharply only a tiny genetic slice survives. And yet, it's that slice that tells us the most. When researchers studied the Lacandan Maya of Chiapas and the Pocomchi of Alta Verapaz, Guatemala, they found something haunting. The DNA wasn't erased, it was frozen. 98.8% of Lacandan individuals share the same maternal lineage. Haplogroup A. Their genetic diversity score? 0.025. That's not normal. That's not evolution. That's insulation. A direct result of centuries of forced isolation, cultural cohesion, and survival at the edge of extinction. But the real twist? This bottleneck didn't erase the past, it preserved it. Trapped inside these ultra-narrow gene pools are lineages so ancient, so rare, they don't show up even in other Maya populations. And one of them matches a haplogroup more commonly found among indigenous Arctic peoples. Thousands of kilometers away, entire ecosystems apart. It's not a metaphor, it's in the genome. So now the question changes: what buried event, what unknown migration, what ancient thread connected the forests of Mesoamerica to the edge of the ice? It begins with a whisper from the ice, a whisper echoing in the blood of the Maya. Because deep inside their cells lies signals that bridge continents, oceans, and centuries of distance. Scientists found rare GM-AG and GM-AT haplotypes in Maya populations, variants almost identical to those in Na-Dene, Eskimo Aleut, and even Samoyed Siberians. archive.org+10n.wikipedia.org+10n.wikipedia.org+10. This isn't hearsay, it's a genetic link. So how did a fragment tied to Arctic peoples end up in Mesoamerica? We're not looking at trade goods or myths, we're looking at genes. This leads to two jaw-dropping possibilities. 1. These signals are echoes of the original migration, the one that crossed Beringia between 20,000 and 14,000 years ago. A migration that sent people south but left lingering traces north and west, and then disappeared into the forests of Central America. 2. A back migration where descendants from the Americas traveled back into northern homelands, carrying their genes and their identity northward again. Something recent, something unexpected. And then there's haplogroup A2, carried by both the Maya and Andean peoples of Peru and Bolivia, despite being separated by over 4,000 kilometers. If that isn't a continent-spanning genetic bridge, what is? It becomes more curious when considering HLA immune genes. These haplotypes didn't just travel, they fused. When Maya immunity genes resemble those in Siberia, it forces us to ask, did they evolve under similar pressures? Or did the genes themselves trace migratory trails we're only now glimpsing? So yes, we inherit blood. But what about belonging? Because DNA doesn't just record ancestry. It carries intention. It tells us why we belong to a story that spans more than a forest, more than a civilization. It spans a continent. And now, the implications are becoming urgent. Because modern medicine is turning its gaze on these ancient codes. What if these rare haplotypes, these shared immune signals, hold both the key to resilience and the seed of vulnerability in Maya bodies today? Are we about to discover that an ancestral heritage stretching from the Arctic to the Andes could be both a gift and a warning? It starts with an echo-ancient survival code trapped in the blood of millions today. This isn't legend, it's genetic fact. The Maya carry a mutation in the SLC16A11 gene. A small change, inherited from archaic Neanderthal ancestors, nearly 50,000 years ago. And today, over half of indigenous people across Mexico and Central America share this same variant. For millennia, this gene was a blessing. In a world of famine, hard labor, and unpredictable harvests, the mutation meant you stored more with less. It was evolution's insurance policy. Efficient, durable, powerful. But the world isn't the same. Fast forward to the 1972nd and 1982nd when processed foods and sugar floods reached rural Maya towns. That once helpful metabolism turned on its own body. Obesity, high cholesterol, type 2 diabetes now hitting Maya communities at an epidemic pace. Studies from 2015 onwards show indigenous Maya adults with SLC16A11 have a 30–50% higher risk of developing diabetes. And with low diversity and other metabolic genes, thanks to centuries of village isolation and endogamy, their bodies lack the genetic backup that wider populations might have. So, what happens when a survival mechanism becomes a vulnerability? Modern science is racing to find out. In 2024, a team sequenced a complete Maya genome from a person with known deep lineage in Quintana Roo and Chiapas. First of its kind, first high-heritage Maya genome in modern research. Now, geneticists are digging for hidden clues in that sequence. Are there protective mutations we never saw? Metabolic safeguards? Or deeper Neanderthal-derived codes that balanced resilience with risk? Because understanding this genome could reshape health outcomes not just for Maya individuals, but for all communities carrying this ancient inheritance. And the next question looms larger than ever. What if that genome doesn't just explain past resilience, but points to new ways to heal from inheritance? Could buried in those sequences lie a blueprint for future survival? Because that genome isn't just a relic, it's an active, living record. And now, a new data point has shaken everything we thought we knew. Emerging from the Y chromosome evidence, those strands passed from father to son, a lineage appeared that doesn't fit. No match in modern Siberia, no link to known Beringian waves. It's different. This rare Y DNA branch shows up in a handful of Maya men today across Chiapas and Yucatan, but its origin story lies in a time and place older than the first land bridge migration. It points to a movement that may have occurred before 15,000 years ago, before ice, before the glaciers receded, before Beringia was even walkable. Yet here it is in the living Maya. This means the story isn't linear. It's layered. We have clear signals of Chibchan southern ancestry, Arctic-covered mitochondrial echoes, Neanderthal-derived metabolic genes, and now, a Y chromosome rooted in a prehistoric migration we've barely begun to map. Every discovery unfolds another mystery. 9,000 years of continuity across Yucatan, Chiapas, and ancient Copan tells us one story: Maya resilience. But this anomalous Y DNA tells another. A story that precedes the Beringians, that ignores modern maps, that whispers of human journeys still hidden. It asks us, what ancient footsteps crossed continents, leaving a single strand of DNA in the blood of Maya fathers? And more importantly, what will the next full Maya ancient genome tell us when it's sequenced? Because if this is just the beginning, the most explosive chapter is still unknown.